We will now wrap up this module by discussing different hybridization assays. Although genomes vary slightly from person to person, it seemed reasonable to try to establish a consensus human genome sequence. The chancellor of UC Santa Cruz, which was one of the lesser known of the 9 University of California campuses, wanted a prestigious project to enhance UC Santa Cruz's reputation. There were existing multi-million-dollar projects in physics and astronomy at other UC system schools. Therefore, in 1984, he proposed to sequence the human genome. After much debate, the Human Genome Project started in October of 1990, which was funded by the Department of Energy and the National Institutes of Health. So we have seen how Mendelian genetics explains transmission of genes, but sheds no light on how genes create cellular and organism phenotypes. Computational biology is a field that attempts to use the genome sequence to ascertain function of genes. Due to the Human Genome Project, hybridization assays were and have continued to be developed to more formally study the association between genes and phenotypes. These hybridization assays leverage the biological property that two single-stranded DNA molecules whose sequences are complementary to each other will exhibit a tendency to bind together to form a double-stranded DNA molecule. This process is called hybridization. Even when the sequences on the two strands do not match perfectly, as long as there is sufficient similarity, it is likely that some base pairing will occur. The tendency of DNA strands of complementary sequences to hybridize preferentially is exploited in hybridization assays. First, a probe consisting of a homogeneous sample of single-stranded DNA molecules, whose sequence is known, is prepared and labeled with a reporter chemical. An immobilized target, usually a heterogeneous mixture of single-stranded DNA molecules of unknown composition, is challenged by the probe. As the probes will hybridize only to sequences complementary to its sequence, DNA sequences in the target that are complementary to the probe DNA sequence can be identified by the presence of reporter molecules. Traditional molecular biology research followed a one gene per experiment paradigm. However, with the Human Genome Project, it was imperative that the entire genome, that is, all 22 autosomes and the 2 sex chromosomes, be sequenced and coding genes in their positions identified. So the Human Genome Project spurred industries to massively parallelize the old one gene at a time assays. That led to the advent of microarrays and subsequently high-throughput sequencing. Thus, research has moved from a one gene at a time mode to thousands of genes per experiment. This, in turn, allows us to study how genes jointly function. We will now discuss the initial technologies for which there were two main approaches for fabricating microarrays. The first was deposition of DNA fragments, which were PCR amplified cDNA clones, and therefore the sequence does not need to be known. This is known as custom spotted arrays and is seen in the image on the left. The second was in situ synthesis, where the sequence of each probe must be known, and the known sequence is deposited on the surface by photolithography, which underlies the Affymetrix gene chip as seen on the right, or by inkjet printing, which underlies Agilent, or by electrochemical synthesis, which underlies ion torrent and ion proton. Let's first discuss the custom spotted array technology, often called cDNA arrays, which was the earliest microarray technology that was developed. Here, PCR amplified products, or probes, are spotted into wells so that each well represents a unique PCR amplified product, or gene. These wells containing a probe are often called spots. To study a given sample, you first need to extract RNA from the sample of interest. When assessing the amount of messenger RNA for individual genes in a given sample, poly-A tail selection is used. Recall that organisms have a poly-A tail added to the end of the mature messenger RNA before it gets exported from the nucleus into the cytoplasm. Knowing that a poly-A tail is at the end of each mRNA of interest, poly-T, that is a string of T bases, which is complementary to the string of A bases, is used to pull down these fragments with poly-A tails. Thereafter, the RNA is reverse transcribed into complementary DNA, or cDNA, because RNA is liable. In other words, it easily degrades. Afterwards, the cDNA is amplified so there are lots and lots of copies of each gene. Often, a label, such as a reporter dye, is added, and then the cDNA is denatured to form single-stranded cDNAs, which are hybridized to the microarray. Notice here that the custom spotted array is a competitive assay, so both the sample of interest, the experimental sample, and a control sample are hybridized together. Because of the competitive hybridization process, these two distinct samples are labeled with different reporter dyes. Often we call these green and red. In this ideogram, the sample of interest, or the experimental sample, is from a tumor and is labeled red. The control sample is from a normal tissue source and is labeled green. These two samples are combined in equal amounts and applied to the surface of the array. cDNAs that are complementary to the probe spotted on the array will hybridize. After hybridization, the surface of the array is washed to remove unbound material, and then a fluorescent detector is used to measure the relative abundance of the transcripts in the sample. In this particular case of custom spotted arrays, spots that are red represent genes that are expressed in the tumor sample, spots that are green represent genes expressed in the normal sample, and spots that are yellow represent genes that are expressed by the normal and the tumor sample in equal amounts. Next, please watch the YouTube video that illustrates custom spotted arrays.